Bridge nav, 30 meters, 180.
Front row, yeah, we're looking to be on deck around 4 p.m., so we should shoot for a pulling off the surface in a little over an hour.
Sounds good. Would think of some very innovative things for the Blue Water Watch.
Yes.
Here's another question in the chat. Is it possible that organisms could be hiding due to the noise of the ROVs?
Yes. There are a lot of fish that we're probably not seeing, or maybe not a lot, but there are probably fish and some invertebrates that are hiding. There's been some work done comparing what's the behavior of fish with just things like still cameras and feeding stations and ROVs, and there are some fish that do actively avoid the noise and lights.
Is that a poliopogon?
I think it is.
It's got that thick base.
Yep.
Go ahead and start a zoom there, please.
For the biogeographic boundary on California, it's Point Conception, not Point Reyes.
Got a few brittle stars hanging out.
Come a little wide there.
They almost look like they're embedded in the sponge instead of just sitting right on the surface.
Yeah, I was thinking that. That's how I look out on a couch.
That's how you look on the couch?
Yeah, just slowly becoming embedded in it.
So instead of having a potato day, you had a sea brittle day?
Yeah.
Roger.
I got a question for Kevin. So this ferromanganese crust is basically a kind of almost like a wet blanket thrown over all the interesting rocks that you're more interested in studying. Is that aggravating, or do you enjoy the challenge?
No, it aggravates me.
Okay.
It fills me with a deep hatred and rage of ferromanganese. No. I mean, it's very annoying because there are very specific rocks I want. So instead of being able to just kind of target grab things, you just kind of have to get more higher end values and hope that one or two of them meet your criteria. So it is kind of a bit less convenient, and it also hides a lot of the structures and, you know, detailed kind of identifying characteristics. That being said, ferromanganese is really cool and it's valuable. It gives you some really good long-term oceanographic evolution trends, and there's cool microbiolomes. Can we have a zoom on that cool-looking anemone?
Yeah.
And so I very much respect ferromanganese.
There's got to be some kind of specific term that is refers to an emotion of annoyed respect.
Yeah.
Go ahead and zoom on in there, please.
I'm sure the Germans have it. And Germans in chat, if you can send it along, please. That one's nicely colored, little starburst.
Yeah. These overhangs are cool.
Yeah.
I'm almost impressed by just how much sediment can sit on a nearly vertical surface in the deep sea.
When you get that boitrudal texture, it kind of catches it and helps coat it.
Is that predatory tunicate down there?
Yep, that's a predatory tunicate.
Wait,
Bridge nav 30375.
Oh, good eye. That was
Yeah.
hard to see.
Yeah, I didn't see that either.
Put a push out in there, please.
I love tunicates.
These are cool.
little mastermind sock puppet.
Yep.
Mastermind. That could be a cool metal band, Mastermind Sock Puppets. Very eclectic genre. Go ahead and look, I'm a little wide.
You know how skates wrap themselves around their prey and
Right.
electrify them?
Uh-huh.
So the name of a good ska band could be Electric Burrito.
I'm sure there's a garage somewhere in Southern California with electric burrito.
So the giant California electric skate actually has a really great scientific name.
What is it?
I'm gonna double check it right now. They're also hilarious, and they will come after you if you're scuba diving.
Ooh.
Is that the same
In a
thing as
hilarious
torpedo
way,
ray?
or?
They
You know how it's hilarious when you die deep in the ocean? Cause
Dark today. Cool.
They are Torpedo Californica.
Oh,
The
that is a
torpedo
cool
ray.
name.
That
That's
is
an
a cool
awesome
name.
name.
And they're torpedoes because they come after you.
They're very electric, and yeah, small ones have been known to occasionally try to sit on divers.
Oh.
Wow.
And, apparently it gives you quite the jolt.
Oh, yeah.
You can feel your fillings.
Now I want a California style burrito.
French
I've had
fries
one follow
inside?
me.
What is
French
that?
fries inside.
Yeah, steak,
Yeah.
French fries, avocados, and lots of cheese.
That sounds awesome.
Hoo-yah.
We have a ways to go till we're back in land, guys.
Oh man.
And I'm not sure what you'll find in the way of burritos in Honolulu.
No.
It's true.
I had a torpedo ray follow me once on a night dive. It was kind of came up out of the deep and followed me along, and or met my partner and I, and we just sort of turned around and kept looking over our shoulder. And it was sort of like a little puppy just sort of following you. So it wasn't like aggressive or anything.
Right.
It was just like, hey guys, what's up? You have lights.
Like, curious.
Yeah.
Mm-hmm.
But I'm glad I knew what it was. 'Cause otherwise, I probably would have tried to touch it.
It's a really good lesson in why, when you are scuba diving, you really try not to touch the marine life.
Mm,
It's a good lesson at all times when diving.
very true.
Or just out in the wild. I wouldn't touch the wildlife either. Wild horses or anything like that.
Yeah, I have this funny story. My childhood friend, their family went to, I think, Zimbabwe or somewhere in Africa, and my friend, she was like a little kid, like a toddler, and she loved like picking up lizards, so she like picked up this lizard, and they were at this like
Is that
national park,
umbaloula?
and the guards were like freaking out because it was
Yes.
a poisonous lizard.
Oh.
Oh, no.
Yeah,
Was she okay?
she was okay, she just dropped it, but just the fact that she was just so open to just picking up lizards and random animals in the wild.
Yeah.
Yeah. Ugh. Well,
I have a question about allergies. Do sea creatures have allergies to like marine snow?
That's a great question. Fine.
I really hope not, since most of them depend on it for food.
Yeah.
Okay. But you know how sometimes, I was just wondering if we knew of any cases of such a...
Not to my knowledge. I don't think they have the same kind of... even the phytoplankton don't really do the like pollen thing.
Oh,
Deep philosophical questions. Do fish sneeze? Yeah. That's a good question. I was like, oh, I wonder.
If a fish sneezes deep in the ocean and no one's around, do you have to say Gesundheit?
I don't know if it's important, but someone was just pointing at your voltage gauges on the camera.
yeah, I think that was Josh.
Starting to get some of these stalked red crinoids now. Still plenty of these green yellow ones floating around. Two different types of crinoids. There's one on the left is stalked and stationary. The one on the right is a feather store, and it can float around, move through the water column, resettle.
Not just float, actively swim.
Yes.
Mm.
It's a nice looking coral. Can't tell what it is.
And if folks at home want to see a swimming crinoid, it got included in a recent batch of highlights that if it's not up and posted yet, it will be soon.
Go ahead and do a quick zoom here, please. Hemicorallium.
Bridge
Yep.
nav.
Yep.
Nice hemicorallium.
30165.
Pull wide, please. Yeah.
So I got a stick right in the middle.
It's a sea pen.
Oh.
Probably Balticina, but it might have been one of the either protoptilum or stachytilum as well.
I was going to say that.
Always remember, those words have extra P's.
Just for Kylie. All right, we're gonna do a pilot swap here, guys.
Sounds good.
Chris, you wanna do a DVL reset there?
Yeah.
It holds. Whoa. Oh, sorry.
Here's a question in the chat. Do we know more about immune systems of deep sea creatures?
I think we know very little about the immune systems of deep sea creatures. A lot of these animals, we're just learning a lot about their basic biology now. One of the reasons it's important to take samples. So that's some of the kind of information that we can get from looking at things like the metabolomics projects that are going on and some of the transcriptomics projects. So information is going to start to come out, but that's one of the reasons it's important to take samples, is to learn that kind of information.
What are metabolomics and transcriptomics?
So metabolomics is ways of measuring the metabolism, so how an animal is producing energy, looking at basically the chemistry that's going on within its cells. And transcriptomics is measuring what genes an animal is turning on and off in response to its environment or a specific stressor. So the collection we made right at the end of the last watch, where we were collecting that feeding triad, they're going to do transcriptomics to see how the coral is responding to the predation event of the sea star, what genes it's turning on and off in response to that stress.
Thank you. Follow-up question: when they're how can you what's the product that they're looking for for the transcriptomics? Are they looking for a protein specifically, or is it the mRNA that
They're
goes
looking
for the protein?
at mRNA.
Okay.
So there'll be you can extract the mRNA and then sequence that. It's called high throughput transcriptome sequencing. Because the mRNA in most animals has what they call a poly-A tail. So the end of the strand has a bunch of A's, so you can bind to that and selectively take out just mRNA and then sequence that, and that tells you what genes are being turned on and off. And you can measure the relative counts, how many counts of each gene you get, and that will tell you kind of which genes are being expressed more or less than others.
Cool. Just by the relative amount of mRNA that's
Bridge nav 30165.
Yep.
trying to think of an analog for that to kind of to use. I'm explaining it to my own students, and I'm guessing like if you took a blood sample from me like right before dinner time, when my body is like ramping up its enzyme production for all the food that I'm about to eat, is that something that you could say like, wow, Tim really eats a lot, or is that the sort of inference that you could make?
It's less of a kind of, you could do that, but what would be more informative is to do that a couple times and then make you skip, say, 3 meals and then take another blood sample and compare that to the normal, you just ate dinner, and you'd see what genes were turning on and off in response to, say, food stress. So I actually did a lot of this for my graduate work. It's called functional genomics, where we were working with a small bait fish from the Gulf of Mexico and measuring their response to low oxygen. So we'd have control fish that are just happy in a normal tank, and then normal other fish that were collected from the same spot as them that we would expose to low oxygen stress, and then measure what genes were turning on and off in the control fish versus the fish that were exposed to low oxygen.
There's an umbaloula.
Yep.
And some crinoids.
Back row is to verify you guys are satisfied with this depth contour.
It's a little bland over here, but let's see. I'm wondering if we should go more onto land or further back. I think further back might be too hard. So yeah, we can maybe start heading towards waypoint 9, see if we see something different.
Waypoint 9.
Might get some more fish in the flatter regions.
Apparently on the descent last night, they went through quite a few fish, more than they'd seen on any previous dive. So maybe on our ascent, we might get some of the same.
Cool.
Very cool.
Bridge nav, 30 meters, 220.
What kind of fish were they?
They didn't say, just that there were many of them.
They sound like myctophids. Small, difficult to identify, numerous, up in the water column at night.
I mean, I would be down for a big cloud of myctophids. I do love some myctophids. More hatchet fish would be good too.
Oh yeah, hatchetfish are cool. I spent some time learning to identify myctophids as part of a college program. The Sea Education Association does oceanography from tall sailing ships, like big schooners and brigantines, I guess. Yeah, one of the things that they would do is do a net tow at night, around midnight, and I was spending time looking at the fish that we collected the next day. And myctophids are little guys that have, little guys and gals that have little photophores, little, kind of like little pockets with bioluminescent algae growing in them.
Cool.
So based on the patterns of those little pockets of light on the underside of that body, that's how you can tell species apart. And there are many species of myctophids. It's one of the things I was really hoping to see when we came out here.
They're one of the things we very frequently, when we run these deep or kind of midwater samples for environmental DNA for fish, they're one of the common species we'll pick up.
Once we get to these flatter contours, we can increase the speed from 0.2 to maybe 0.3, 0.4, if that would be desirable for you, Kevin.
Raj, that'd be great.
See, the Raj is catching on.
Raj.
Raj for life.
Who is the Roger of which everybody is speaking?
Kylie.
Wait.
Well, it's Roger Wilco, right? Who was Roger Wilco? Was that an actual person?
Sounds like a Jeopardy answer.
Who are the pilots here?
Was that?
Oh, no, sorry. Oh, the small plane pilots.
Tim, I have you at +6 and you're still whispering.
I will crank it up. Yeah, Dave's on right before me, and I think he likes to be in the corner, kind of quiet, unassuming,
Yeah.
anticipating, not really. I don't hear him much on SPL when I listen in, so.
Yeah.
So I have an answer for your Roger question.
Oh yeah.
So it's the spelling alphabet. Is the R is Roger, and what that means for why it's used is because R in radio communications means received.
So that must be the old phonetic alphabet,
Yes, I think so.
because now it's Romeo.
So should we switch to Romeo?
I don't know. I think they I think they switched that sometime just after World War II. I'm not entirely sure why, but
Maybe it became too common as a name.
well,
Yeah, I am.
Apparently it dates all the way back to Morse code.
Okay, then, if that's received, then Wilco is,
Just wagging my tail, you know?
will comply?
I am. I am.
Will cooperate.
According to the internet, it does mean we'll do what you request.
Got a few little corals hanging out on top of a boulder. There's more over here. This big white brittle star to the left, or not brittle star, but sea star.
bridge nav 30225.
It's like a big crinoid with his stalks up, straight up into the water column. I mean, unstalked, sorry. Got some very big blocky boulders over here. Quite interesting. I don't even see on high pack where they could be from, so. There's a little fish.
Apparently, with the Roger versus Romeo, it did used to be Roger, and at some point it was changed to the current Romeo. That was adopted in 1957.
Which also happens to be the U.S. geophysical year, or International Geophysical Year.
Coincidence?
Ooh.
Oh. Oh, geologists are always at the heart of a conspiracy theory.
I thought that was physicists.
Oh, yeah. Is that another sea pen on the left? No, that's a coral.
Yeah, I think it was a big, hemicorallium.
Yeah, I was seeing it from the side. There's that fish.
I think that was a cutthroat eel, but didn't really show us its head. I think straight ahead we have a crinoid, a traditional crinoid pretending to be a sea lily.
Is that the one on the stalk?
Yeah,
What's up, Jess?
it might really be a sea lily.
Yeah, that would be weird if it's yellow.
Yeah. Bridge nav, can we increase speed to 0.3 knots?
Kevin, could you explain a little bit more about how these rocks were formed? I know you said this might be limestone.
Yeah, so the on the flatter part of the seamount, so a lot of these rocks probably represent ancient stony reefs, where limestone secreting corals and actually since it's Cretaceous, mollusks, likely built on top of themselves, generating a large, basically carbonate-based structure. Eventually this seamount kind of subsided too fast. Can we have a zoom on that fish when you get a chance?
Yeah. Right behind the umbaloula?
Yeah.
Raj. Romeo.
And drowned it out.
Go ahead and zoom. Is that a halosaur?
Yep, that's a halosaur.
Saltfish. Salt dinosaur?
And we've got its big xenophiophiore right underneath that halosaur.
Where? I don't see it. Oh. It looks different than the other ones we were looking at.
Yeah, this is probably a different type.
Can I get just like a maybe partial zoom on that? A little far. That's good. Oh, wow. Okay, come on. I'm fine.
Yeah. Bridge nav, 40 meters, 225. Oh, 0.4 knots. Yeah. I even put in a bigger move in anticipation of a little bit faster speed, and then didn't talk about the faster speed.
Oops.
Yeah, everything looks good. Correct.
So do the actual sandcastles where the xenophiophiores are making serve a purpose, or is it a residue of its purpose, of it leaching out the sand?
You know, I don't know. I suspect they're just... I don't know. I suspect that they it's just a sand coating on the outside that doesn't serve a specific purpose other than maybe a little bit of armoring.
There's a really flat wall, probably carbonate building on itself. It's hard to tell though. Got two different textures, a little more bulbous on the center and then very blocky on the right.
Yeah.
I'm guessing these are big blocks from the collapse of the atoll wall up ahead, but that wall is kind of far.
So inside these was where we would find our slacker corals.
Yeah. Yeah, it seems like there's two. I mean, right here we're on like the first of a flat spot, and then if you go like 100 or 200 meters ahead, there's a second steep wall. So I think it might be 2 generations of reefs.
So would you consider that a paleo shoreline?
Not here, and probably not. You can actually see paleo shorelines. And actually in this geo, you can kind of see a little bit of paleo shorelines. It's kind of stepped on the flattest part. Well, not up to that, but I don't know if I would consider this a paleo shoreline. I don't know if this was ever above water. It's hard to tell.
These are cool rocks.
Yeah. Very smooth.
Smooth and just so big boulders in a line, like, you know when you stack them at the beach?
Yeah.
Sort of like
Like a jetty?
toppled over, yeah.
Here's a jetty built by the ancient dinosaurs.
Trying to start a new dinosaur conspiracy theory.
Yes.
Pyramids are built by dinosaurs.
They built jetties.
So we have Ken Sulak, a world expert on deep sea fishes, in the chat, and he said that halosaur we were looking at was Aldrovandia phalacra, if I'm pronouncing that right.
That's a pretty name.
Yeah.
I wonder what it means.
Bridge nav, 40 meters, 225.
There's a question in the chat. Could the ocean floor look different if there were less ocean acidification? If so, what would the ocean floor look like?
Sure.
That's a good question. This deep ocean, if we're talking about recent kind of anthropogenic-driven acidification, this deep ocean probably looks the same as it did in the past, because those effects haven't really got down here. But you see it very dramatically in the shallow waters where you have coral bleaching events, where you had these colorful, beautiful, diverse coral, reefs that are starting to turn all white as most of the species die off. So yeah, it has an effect. If the acidification here changes or decreases, it could affect how much carbonate sediment can be deposited. And that will change, how sedimented this region is. So we're kind of in an area where you don't really get anything from the terrestrial realm, just tiny bits of windblown dust, but even here it's actually quite small. So that's why we have very little sediments. But if we decrease acidification and carbonate can last longer, you can potentially build up some of this marine snow more. It's like a crinoid on a sponge. Or is it behind the sponge? No, it's on it.
On it,
So
yeah.
I think it's 4
Two of them,
crinoids on the
yeah.
sponge.
Yeah. New sitcom on CBS.
Oh, we got a little coral right there too.
We have a zoom on the coral?
That's a
Sure.
That's a black coral, but I'm not 100% sure.
Zoom.
Yeah, that's a little black coral.
And a squat lobster.
Yep.
Okay, come wide.
Yep, Steve is confirming that that's the little stauropathes, and we collected one of those just a couple of days ago.
Oh, good.
I thought so, but I couldn't remember exactly when. All the dives are starting to blur together.
Totally. Umbaloula. Under my umbaloula, lula, lula, hey, hey.
There's an anemone. They seem to like the kind of bottom side of the rocks.
You get a zoom?
So why do some of these stauropathy crinoids fold their feathers versus just having them sprayed out? Do we know?
I have no idea. My guess is it has something to do with either feeding or being done with feeding and
Come wide.
getting out of the current.
Oh.
Do we know the life cycle of crinoids?
My guess is it's very similar to all other echinoderms, where they will be basically broadcast spawning gametes, and then they spend a while in the plankton, and then metamorphose and settle out.
So for a coral, when you have these gamete events, are we talking thousands, millions, billions? How much of these potential seeds are sent out? Do we know?
Corals can further complicate some of things because there are some that are broadcast. Most of them are broadcast spawners, but some of them are what we call brooders,
Got some sponges.
where only one of the gametes gets broadcast, and then they actually brood the larvae as a sub-polyp on the main adult, and then eventually basically bud off that polyp, and you get a lot of local retention that way. But for broadcast spawning, it's, you know, hundreds of thousands of gametes.
Broadcast spawning. I've never heard that before. What does that mean?
And it means they just release their gametes out into the water column.
Oh, okay, okay.
Usually as kind of a mass event where a bunch of them are all doing it at once to maximize the chances that, you know, the right gametes are going to meet. But
Same idea as when, like, the kinds of flowers that are, like, I don't know, maybe they're self-pollinating, I'm not sure, but when they just, like, the wind takes their
seeds?
Yeah, similar, except those would be more like brooding because the flowers that are receiving the pollen aren't leaving the tree. So it would be like if you were throwing the flowers and the pollen all at the same time.
Okay, I see.
Really good example of this is tropical coral reefs. Usually in August, right after a full moon, all the coral species will broadcast spawn, and their gamete packages are large enough you can actually see them. So there's really good like video footage you can find on YouTube of mass coral spawning events.
Oh.
There are examples in fish too. So a lot of fish that will just spawn into the water column and let their larvae develop and float free. That's broadcast spawning. But then you have species like seahorses that will actually brood their eggs.
How long is the gestation period of a seahorse?
I don't know, but I bet we can find out.
Also, does gestation period have anything to do with like lifespan or no?
Not really.
Okay.
So in seahorses, it's 10 to 25 days depending on the species.
That's convenient. It is.
Looks like we're just about going to make it to the waypoint 9 before we have to pull the dive.
Sounds good.
Or at least to that depth contour.
Yeah.
Bridge nav 50225.
There's a little halothorian on the left
Yeah.
and a big crinoid rock.
So we're not collecting halothoriums anymore?
We're still keeping an eye out for urchins, but Liz is pretty
Okay.
happy with halothorians right now.
We've got a bunch of them, right?
Yeah.
Yeah, okay, I see.
Can we look at this sea pen?
Yep. Okay, video zoom. Oh, sorry.
I think this is one of the Protoptylidae that they collected just the other day. Yeah.
You can barely see its polyps.
Yeah, very,
Polyps are very clear.
yeah.
Thank you.
No problem, come wide. Just. Okay, let me just make sure I'm centered on my bridge. Bumped around. Okay.
Ken Sulek just sent us a really great paragraph of information on the chaunacops. It says that these intriguing fishes are also called sea toads,
Oh, yeah.
coffin fishes, or gapers. And only the last of these common names make sense. When a gaper makes a prey strike, the mouth is opened enormously, which creates suction that sucks in the victim whole.
Video zoom.
We may want to sample this, and we can put it in the starboard box with the other one.
Bridge nav, hold position.
Yeah, we did have a request if we came across another urchin, if we could sample one more.
Okay.
So this one will need to be like scooped.
Come wide, I'm gonna set up a little differently.
Bridge nav,
So this-
30040.
So this is going to go into the starboard bow?
Yeah.
Okay.
30 meters, 040.
So starboard bow, we won't be able to scoop nor have suction to aid us, so we'll just grab it with a minute.
Yeah, box E just has a very small rock in it.
It can actually go in with the other urchin in the omega box.
Okay. In the forward bow.
In the
box.
forward box, yeah.
Roger.
Okay, side question. From my understanding, that one's kind of floaty. Can I put it in the E, or?
I don't think they want to pinch it.
I don't think they actually want it in with a rock in case the rock rolls around during recovery.
Okay. Roger that.
Okay. The sense and benders off.
Roger.
Craft power is on. I'll wait on comms before I turn on the valve.
Yeah.
Want me to get it under your forearm?
Yeah, thank you.
I know.
Silly, silly, silly.
It's temporary.
It's a temporary feature.
Just remember you have that rock stuck in your fingers too, so.
Oh yeah, that actually might help us.
Okay, cool.
Maybe it might hurt us.
We won't know until we try.
Yeah, fun. Oh, you want to give me the valve there, Kelly?
Yeah, valve, yeah, sorry. Raj, come on.
Okay.
Okay.
Cool.
I wonder if you'd grab the slurp, it'll push the rock out.
I don't know. Let's see, I'm gonna probably just slurp it, and then we can dump it in that way. Science.
That sounds fine.
Cool. Let's do this one more time. Chris, you want to keep an eye on our sonars, please?
I can do.
Oh, yes, it got the rock out, which is good.
That's great.
Unintentionally. You want to look a little down, please? Thank you.
Let me know when you're ready for any suction.
Sure. Oh, I'm having such a hard time with this guy. Stand by.
Yeah, Raj.
Cool.
Okay.
I'm gonna go ahead and look a little up there. Chris, you're backing the ship up, correct?
Yeah, I am backing the ship.
Okay. Sorry, I was pulling up on the winch a bit.
Okay.
All right, go ahead and give me some suction, please.
Okay, you have 30%.
Bridge nav, 20 meters, 040.
Might need more because it's big.
Go ahead and push on in there a bit, Tim.
Tilt down a little. Okay, more. It's 40%. Oh. Wait,
Okay. A little more.
Raj.
Thank you. Full wide, please.
Okay, ready for tool tray out?
Yeah.
Coming out.
Okay. And look a little down.
Yep. Looking down.
Gonna look a little up actually now. Sorry.
No problem.
Thank you.
I'll just try to follow it in with you. Oops. Oh, sorry.
It's okay.
Touchy.
Thank you. Okay, this is a awkward one.
Yeah. Whenever you're ready, I can decrease section.
Yeah, go ahead and start decreasing now, and I'll see if I can just waft it in.
Okay. I'm doing it in
Oh, increase it again, I guess.
Raj. Okay, got that back. Ready?
Stand by.
Okay.
I can't really see.
What? Yeah.
Gonna look up a little bit.
Sure.
That's great.
Okay. A little down.
Yeah, a little down. All right, go ahead and kill section there.
Okay, section is off. Well.
Well, we don't like that, now do we? All right, we're gonna throw this guy out here for a sec.
Sure, yeah. It's all set.
Go ahead and look a little bit up.
Is moving towards. Should I back up?
Go ahead and start closing the bin,
Okay, coming in.
and then look a little down.
Looking down.
Maybe that'll actually get his tube feet out of there.
More?
Oh, he's climbing. Go ahead and close it all the way.
Raj,
Yeah.
closing. Get in the bin. Not all the way, right?
No, keep going.
Okay.
Now jostle it open.
Open.
We're gonna see if we can stun him. Now close,
Closing. Get in the bin.
and then
Get in the bin.
open. Yeah. Again.
He's just so big.
Keep doing it. I think you're getting somewhere though.
Yeah, I do too.
I got a twitchy wrist here, so.
Okay.
Putting the shoes to get
Uh-oh.
used. Oh no.
Oh.
Get
Okay,
away.
now go ahead actually and try lifting off a wee bit.
Okay. Take the locks off. Come on.
Okay, I think this one actually is gonna crawl away.
Don't you dare.
So we don't want to jeopardize the other ones in the bin.
Yeah.
Oh, in the time to go straight up if you want to see if it'll stay on the deck.
Okay.
All right, go ahead and I'll close up the bin.
I can do that, yeah. I got the in.
Son of a gun.
Put those little urchin shoes to good use and ran
Yeah.
away.
How about we try this? How about try backing up? Try the Josh special, I suppose we can call it.
The jostle.
Yeah, go ahead and
Okay. Okay, backing up. No.
yeah. You have stick lock on right now.
Oh, Raj, thank you. I was like,
There we go. See if we can just get him out of there.
turn the other way. Goodbye. He's so big for.
Nice.
Get out. I'm going down.
Good. There we go. Nice.
Okay.
All right.
Coming up. I can get him out of my brow. Let me look up a little. Okay. Jeez.
All right.
Open the bin.
Yeah, we don't want to jeopardize the other one that's already in there, though.
Raj. Okay.
It would be cool to do another slam dunk, but I don't think that would be good for the other samples.
Okay. Maybe we just try suction again?
Yeah, yeah, I'm just going to grab it with my hands.
Okay. Okay, all right, sorry. I got the suction dangling there.
Yep. I'll get that sorted out right now.
Okay.
There we go.
You want to try one more time with a suction so that it doesn't puncture him because he's so big?
Nah.
Okay. He's really fast.
Yeah. Go ahead and look over. Great force is at 2.
Okay. So
Go ahead and push on in there, please, Tim. That's good.
yeah, I'm gonna get closer. No, you're good.
Here, look a little up. Yeah, now I'm at the extent of my joints. You want to get a little closer?
Okay, come on.
All right. Nice. You look a little down?
Yep.
Kick it out this way off the rock.
Nice. Okay, coming out.
Roger.
Looking down.
Ah, the grip.
No problem.
Oh, sorry, guys.
Their shells are surprisingly soft.
All the way out.
Yeah, all the way out.
Okay, coming in.
Sorry, buddy.
Okay.
All right, Chris, what's going to be our recovery heading?
090.
Roger. Do we want a last eDNA sample here too?
Yeah, let's go ahead and fire the last Niskin here right as we take off.
Data, Data, what was the sample number here?
135.
135.
And we'll have Kylie lift off the surface a bit, yeah, just
Yep, that sounds great.
Okay.
great.
And should I turn to 190 or is any heading okay?
Any heading's okay.
Okay.
Oh, sorry,
Can I
for
get the wrap
recovery.
out or first?
Yes.
Raj.
Do you mind if we grab this Niskin though? So we'll
Yeah.
just get out of the thruster wash.
Yeah.
And while you do that, I'm gonna do some maintenance here.
Okay. Get out of the thruster wash.
So the jaws don't close all the way right now. Is there another rock in it?
No, not that I see, no.
Cool. So this is full closed.
Maybe all the way open and see the... Oh, Raj.
Maybe the rock jammed something in there.
Maybe.
Cool, that'll have to be checked.
Okay.
It's floppy, look at that.
Yeah.
Cool.
Yeah.
Wow. Cool.
Just one side of it, yeah.
Both sides.
Okay.
Do you see it flopping open?
Yeah, yeah. It's all off kilter.
Okay. All right, we'll go ahead and get a Niskin sample here.
Okay.
Gonna look the
Let's
broken
hop
jaw.
Eh.
Cool.
Sea urchin's revenge.
So I think you want the green one.
Green on Raj. look over here.
You got it. Broken jaw and all.
Popped.
Good.
All right, and we're 2.5 meters from the bottom there, Meredith.
Oh, perfect. Thank you.
Yep. Wow, that's so crazy. Cool. Yeah, you look well down.
So there's a message in the chat about eDNA. Is there clearly distinguishable DNA that we find in eDNA samples that we don't yet know what the where it belongs, like which organism it belongs to?
Yes, so we'll capture everything in the local area, and some fraction of that will actually be stuff that's not yet in our database. We can still sequence that, and that's a really good signal to us for searching for new animals in the area.
090, right? So I'll actually go to 090, because that will be our recovery heading right there, Chris.
090, correct.
Great. And then you'll go to the reciprocal of 270. Yep, that's looking good.
Thank you all for tuning in for the sea bottom. So we're going to start our ascent towards the surface.
Yep, yeah. Once we get stretched out, we'll just start going up, and then we'll increase the z-bias there.
If all goes well, we're going to transit to the south and try to dive on another seamount called Wetmore. Fingers crossed. So what is everyone's favorite cheese?
Cotija cheese for me.
Oh, good choice.
What kind of cheese is cotica? Is what you said?
Huh?
What kind of cheese was that again?
Cotija cheese? It's like a dry cheese.
It is the key that makes elote the most delicious food in the world.
It's
If
true.
you haven't had it, imagine like a feta, but a lot better.
All right, ready?
I like a good Gouda.
I would say it's situational. It depends on what I'm eating the cheese with. So like a really good, like, you know, if I'm just eating like a cheese and crackers, like a really good solid sheep's milk cheese is really tasty. But like if I want to bake with it, then like a queso fresco is more my jam.
Yeah.
I don't think I have a preference of cheese. I just don't like brie. It's not my thing.
Is it the wax or the actual interior or both?
I don't know. It might be all of it.
I'm going to have to go with brie, actually.
Conflict in the
No,
van.
I was like, I'm going to be honest.
I mean, a baked brie with like apple in it or something. Fantastic. I've made those and then consumed the whole thing myself before taking it to the event. Looking at maybe a 4-hour transit, but we don't know exactly yet this time. To answer the question in chat, sorry.
Yeah, transit to our next dive site.
Yeah, maybe 4 hours, we'll see.
Cool. There's a couple more questions in the chat about the eDNA. So can anything be determined from the unknown DNA, like what phylum it might belong to?
Yeah, we can actually, so we can, assign unknown sequences back to their family usually using a Bayesian assignment tool that we have.
And then how long does the DNA last in the environment before it starts to break down?
It depends a little on where it is. In surface water, no more than about 24 hours. Deeper, probably a little longer than that, but you still get some degradation within the first 24 hours, and then a slower degradation after that.
Roger.
Did we take any limestone samples?
I believe the previous watch grabbed 2 that Rob thought it might be limestone, so we'll see when we cut it open.
Oh, you go.
No, continue.
Oh, would it be possible to find like marine fossils?
Yeah.
Certainly.
Yeah, you're not going to find like big fishes or vertebrates or anything like that. It's going to be microorganisms, coccolithophores, foraminiferous. You might get some shells from mollusks and stuff. And yeah, they're going to be kind of compressed, so they're hard. You can't really separate them out, but you can see them still, and when you cut them, make thin slabs.
And would you be able to figure out what geologic time frame?
Yeah. Yeah, I expect these to be mostly late Cretaceous. So it's hard to say. We don't know how old the seamount is, but somewhere around 70 million, we'll say, to be safe. And I don't know when it finally went underwater and the reef stopped, so it could go into the, Cenozoic pretty easily. I have a question for Meredith. What's the turnaround time on an eDNA analysis?
It'll take me, with these samples, once I get them shipped back to the lab, probably 4 to 6 weeks to get them all extracted.
Oh, that's nice.
And then another 4 to 6 to get the sequencing done. Some of that is just time to get scheduled on the sequencer.
Yeah.
And then and doing some initial data analysis as well. So about 12 weeks.
Okay. That's pretty good turnaround time.
Yeah, and then we have some batches of samples from previous expeditions that we'll be combining with these as well.
Nice. So does your project, is it just everywhere you can get samples in the Pacific, or is it focused on the Prim, or?
It's been kind of anywhere we can get them in the Pacific. So we've got 2 or now 3 rounds of samples from Papahanaumokuakea, different parts of the monument. Got samples from Howland and Baker, and the Phoenix Islands,
Nice.
and Jarvis, and American Samoa.
Awesome. Nice tour of the Pacific, the latitudinal transect.
Yeah, so we'll be looking for those biogeographic boundaries we were talking about earlier, using both the physical samples that Steve is working on, and then pairing those with the eDNA samples.
Cool.
Could you explain what eDNA is again?
Yeah, so when we say eDNA, that's it's a small acronym for environmental DNA. And what that is yeah. It's basically any of the material, mostly cells, that animals shed into their environment. So sitting here in the van, each of us is shedding about 40,000 skin cells an hour just into the van.
Nasty. Go on.
But we can capture those in a water sample and then on a filter on the ship.
Are the Niscombados sterilized in between dives?
They're not, because there's not really any point in doing that, because they're open to the water column all the way to the seafloor.
Do you have to preserve the eDNA samples?
We do. We preserve them in a buffer that we've got aboard the ship called Longmire's lysis buffer, and it's just basically a salt solution that stabilizes the DNA at room temperature for years and years and years. That gives us plenty of time to transport them back to the lab.
Here's another question in the chat. There seems to be a lot of greenish yellow and red color species. Why are those colors so common in the deep?
Red is really, really common in the deep, and that's partially because the water absorbs red light really, really effectively. And so animals that are red, basically to other animals, even if they have bioluminescence to kind of light things up, anything that's red would basically appear black on a black background. So it's kind of an invisibility cloak in the deep ocean.
I have a question for Chanel. Can you tell us about your PhD thesis project and why you decided to betray and stab a geologist in the back?
Oh gosh.
Maybe just the first part.
It's not like that. I still love geologists and geology. Yeah, actually, switching into more of a biology-related field was inspired by my first time at sea. I sailed from Rhode Island to Barbados on a transit cruise and was helping a research team build a flux cam that they were going to use to study phytoplankton in the surface waters from the Amazon runoff. So that was really cool, and I was learning a lot about how surface level microbes can affect a lot about our oceans and our atmosphere. Like it affects so much more than just the fish that eat them. It also affects, the air that we breathe. And it also kind of gave me a lot of confidence to study that. I was really intimidated by biology beforehand. And then I went on this cruise and I was like, I literally built a flux cam to study microbiology. Like I can study that. And then I was also just always interested in life in extreme environments. So I decided to combine those two things and found a project with an incredible advisor where I can study life in Antarctica, which is an extreme environment also affected by climate change at a faster rate. The poles are being affected really quickly. So it kind of tapped into all the things I wanted to study, and I get to study, the carbon contents in the soils there, so I'm not fully moving away from rocks. And now I'm trying to use lipid membranes of these cells that live there to try and understand what kind of adaptations they're making to their cell structure that allows them to live in this extremely arid and extremely cold environment. Because there's so little biomass, so we can't just go collect cells and do cell counts and all these things that people who study surface ocean waters and normal soils can do. We have to study kind of proxies for activity, and lipid membranes serve as that role. So yeah, that's kind of a long-winded answer, but
That's great.
yeah.
Have you been afforded the opportunity to go to Antarctica?
No, but we are in the process of trying to go. Yeah, so the field site that we're studying is called Elephant Head, and it's one of 2 locations on Earth where dry permafrost has been found, which is defined as ground that stays below 0 degrees Celsius for 2 or more years, and it's incredibly dry. And no one has ever actually studied the field site that we're studying. We're the first group who's getting data on it, so hopefully we can make a story about the preliminary results compelling enough to convince them to send us back.
Yeah, it sounds like the ideal analog for Mars.
Exactly.
Yeah.
And, drier places are going to get drier and wetter places are going to get wetter as the climate continues to change. So it's kind of twofold. I get to study an environment on Earth that is similar to Mars, which is, that's awesome. And also, maybe we can understand what kind of adaptations cells might be making as the Earth morphs into this more extreme environment.
Yeah, awesome stuff. You'll get to enjoy Happy Camper School, which is the survival training you have to do as soon as you go to Antarctica.
I would love that.
Oh, we'll see. My wife did it, and at one point she was in a tent, and it was negative something, and she woke up and her whole side of her head was like a big ball, all of her hair was knotted together, and she couldn't figure out why, and it was kind of frozen, but then her tent mate told her she was breathing, and her exhaust would come out of her mouth, and then there was snow back down on her head.
Oh my gosh.
And
wow.
So she made her own snowballs.
I feel like spending 20 years in North Iowa kind of gets me some cred.
Oh, yeah. There were times when she was at McMurdo Station and it was colder in the Great Lakes than it was in Antarctica.
Yeah, I remember those times in Iowa. That's why I moved to Phoenix.
Just only extremes for you.
Yeah, literally. I know. I don't know why I did that, but I have a good time. So that's all that matters.
Yeah.
I think some of the coldest I ever experienced was a windy day in the middle of winter next to Lake Erie. When the wind just rips across the icy lake, it's insane.
It just gets into your core
somehow.
Yeah,
it's not even an absolute temperature thing. It just it's brutal.
Yeah.
Your eyeballs start hurting somehow. Didn't even know I could feel pain there.
There's that famous Mark Twain quote that says something like, "The coldest winter I ever spent was a summer in San Francisco."
That's awesome.
I mean, he's not wrong. The damp will kind of just seep in and get you.
I'm spending a couple weeks in San Francisco after this cruise, and then I'll go back to Phoenix during the hottest season. So we'll see how I respond to that change.
I find San Fran lovely year-round, personally. Phoenix, not so much.
Yeah, that's fair. You probably experience similar temperatures in Vegas.
Yeah. The winters are nice.
They're so nice.
Then it gets really windy in the spring, and then it gets real hot in the summer.
I prefer October and April in Vegas.
Yeah, those are good times. There's the weather right now. Yep, it's currently 110 today there, so.
Ugh.
That is the right answer.
Yep. I'm racking up like $200 electrical bills just to keep my cats living in comfort.
A lot of people do solar power yet in
Yeah, it's
Las
very
Vegas?
common. I'm in like an HOA thing, so I don't know if I can put them on, but I would like to put solar panels. You're seeing them more and more on houses.
The last time I was in Vegas, which was like 2019, April, I was actually really impressed by how much like native plant landscaping, like desert landscaping, was all over town, as opposed to like parts of Southern California, which are a similar desert, but it's all grassy lawns.
Part of that is legality. We're not allowed, since Lake Mead is so low, we can barely water outside plants. But, and then part of that's just, yeah, you should take pride in the local environment. You can make some really cool desert gardens and stuff. But yeah, Las Vegas actually has one of the best water reclamation, actually probably the best water reclamation systems in the world, where almost all of our water is reclaimed, except for things that go into landscapes. So anything that goes to watering lawns or farming, there is agriculture in Nevada, which is crazy. That is all lost. But pretty much everything on the strip, like anything in a hotel or a house, that all gets reclaimed if it goes down the drain.
Cool.
Yeah.
That's some pretty major infrastructure.
Yeah, I mean, it was built to try to last. We'll see how we go, how we do. The city keeps growing every year, which is concerning.
Here's a question in the chat. When we take up our samples, do we know if there are any like animal pathogens that are of concern?
I'm assuming that they mean of concern to people. And typically with a lot of the invertebrates, that's not something we really worry about. They are not a big source of zoonotic disease. If you were to take any like marine mammal samples, so at the Northwest Fisheries Science Center, my colleague Kim works with the killer whale program, and they work on killer whale diets. So they are collecting killer whale poop from the surface ocean. And that you actually have to treat as hazardous materials because marine mammals can carry diseases that can transmit to humans.
I have a question for Tim. If you could design a summer research field trip program for your students and yourself to do anything, what would it be?
Whoa.
Yeah.
Anything, huh?
To a reasonable extent.
Yeah. I mean, I'd love to see the rings of Saturn. No. Yeah, that's a great question. I mean, there's a sort of a trade-off in between getting kind of a broad introduction to like lots of different techniques and possibilities and I guess areas of science, or going like really, really deep into one for. So let's say I'm limited to a certain amount of time, like I don't know what, maybe like 4 weeks or so.
Yeah, that's a good time frame.
Like a month.
Yeah.
Yeah, I mean, an incredible experience would be coming out on Nautilus, of course.
Yeah.
Also because, you know, I don't have to worry about cooking for anybody. So that's a big part of the logistics of it. Yeah, I would, I'd probably do something that's place-based, right? So that we're sort of staying in one general area and really kind of getting a good look at all the different things that support an ecosystem there, or you know, maybe just do something like working from the top of a mountain down to the bottom of the ocean in an area.
Yeah.
So somewhere like Hawaii would be a really cool place to do that, because, you know, like you said, you could follow biogeographic gradients from the summit of Mauna Loa. We could get access down to, you know, underwater, the reefs offshore. Yeah, and try to kind of like tie everything together that way contextually. I think another aspect of it would be to bring in different sort of ways of knowing.
Right.
So not only sort of like Western scientific approaches, but also, you know, biocultural approaches, traditional ecological knowledge from some of the, you know, wherever we were, the indigenous people.
Yeah. That sounds awesome.
Yeah.
Yeah, Yvonne was telling me about on Oahu, the highest point, which the name is eluding me. You hike up there, and there are flowers that you only find on that high point. Like there's wildlife that's just there, so you can really see some biodiversity going from there all the way down to the flanks of Kilauea. Would be awesome. Not Kilauea, Oahu. Good answer. Thank you.
Thanks. That's a great question. So I have a question for you, Kevin. How do I explain radioisotopes and radioactive decay to my 10th graders in a way that they'll understand it?
That's a great question. And I'm definitely not the person to ask, but I think the key, yeah, it's kind of, they have to understand transitions. So the mass and the atom isn't disappearing, the parent. So you're basically having an atom that just changes into another one through time. Because it wants to be the other one, but it takes time, essentially. So you can have certain things like carbon-14. You can have an atom, an element, an isotope that is created actually from solar radiation, cosmic ray interaction. And so it gets brought into the world, but it's not stable, and it wants to go back to what it was. And so that's how carbon-14 works. You have nitrogen-14 in the atmosphere. It gets bombarded by cosmic radiation, and it forces it to become carbon-14. And then it tries, on average, over, 5,700 years to come back to nitrogen-14. And that's common for a lot of things. Some of these things are made when the solar system first made. So when you have this big bang, not big bang, but a supernova event, you create these elements in this massive explosion. And slowly through time, they just try to come back to being something else. I'm sure that was just more convoluted than...
No, that's great. And I was thinking, like, my students will understand, what the main subatomic particles are, like protons, neutrons, and
Okay,
electrons.
good. Yeah. Yeah, so in that case, then you just have an instability where you can't have two elements with the same total mass that are both stable. So essentially, if you have a element like calcium-40, and an element like potassium-40, and an element like argon-40, one of those is going to be the most stable state in nature. In this case, it's argon-40. And so they're going to eventually, through time, try to break down into that stable state. They're going to jump what's called an energy well and release energy to become the most stable they can be. And that's what most of radioactive decay is. Elements trying to become their most stable member. This stability might come by ejecting a proton or taking in a neutron or ejecting electrons, etc. But the end goal is always the same, is the stability within their mass range. Everything wants to be stable.
So then the way you would use that for dating, like for carbon-14, would be you have to know sort of the concentration. It can only be created in the atmosphere, right?
Yeah.
In exposed atmosphere, and then when it's ingested or whatever by an organism,
Yeah.
it's no longer exposed to the atmosphere, and you kind of start the clock. It starts to decay over time.
Yeah.
Right.
So for carbon-14, it's kind of unique. You need to actually know what the carbon-14 value of the atmosphere is through time, because it's not
Right.
steady.
That was what I was just getting to. So
Okay.
how do you do that?
You do it through independent means. So the main ways we have ice cores, where we can actually count the annual layers of an ice core,
Okay.
and then measure the carbon-14 in it, and we can back calculate, okay, what was the initial atmosphere
Okay.
with that. And then we also use speleothems. So these cave deposits, these little limestones that have really high resolution, you can look at those and age date them using a different method, in this case uranium thorium isotopes, and then use that to back calculate what the initial carbon was.
Okay.
So what we create is what's called a carbon curve through time,
Okay.
and you can use that to correct for your ages. It actually becomes a big issue in the deep ocean, because the deep ocean and to the atmosphere has such a different accumulation time that you have to
Sure.
you have to have models to figure out exactly what the initial carbon would be in the deep ocean, and it varies by region.
Right. So when you're doing radiometric dating for geological stuff, that's much longer term and more based on like the formation of the Earth or the formation of the solar system or something like that. So that's a constant basically.
Yeah, you're often looking at things that fractionate the parent from the daughter,
Okay.
and then waiting through time. So for what I look at is argon geochronology. What happens is when you have a magma flow cooling, you lose almost all the argon, and you just have a little bit of initial atmospheric like argon. So argon is a noble gas. It's highly volatile, so it degasses during eruption. But potassium, the parent, doesn't. It stays in that lava flow. So once that cooling stops, now all of a sudden argon can't escape because it's crystalline.
Okay.
But the potassium's still there. So now the clock starts.
Okay.
So you have an event that starts the clock, essentially.
Right.
So that's why it works well for really igneous rocks. But like sedimentary rocks, when they're just you're taking a grain from somewhere else and depositing it in the ocean, there's no direct way really to date when that happens, because the clock never stopped.
Right.
Or never starts.
So, and this is all exponential decay stuff. So, I guess the further you go back, you like have data basically infinitely far back because it's exponential. It never goes to zero.
Yeah.
But I assume you lose resolution at some point, and effectively you can't. At some point, you can't date any farther back.
Yeah, exactly. The cutoff is usually like 5 half-lives, and then once you lose that, you can't measure the parent because it's just. I mean, theoretically, it's never gone, but it is not measurable because it's essentially background. Yeah. But yeah, that's a good observation. Yeah, 5 times the half-life is usually the upper limit on most techniques.
Yeah. So you basically, the older things your error bars get bigger and bigger until you just can't distinguish from the background anymore.
Yeah.
That's kind
for
of
some. That's if you have a limited amount of parent. But yeah, most systems the younger is the main issue. Like I study argon geochronology, we could measure anything from the age of the solar system to about the youngest is 10,000 years now, which is pretty crazy because it's a very long decay process, and we can measure it with such high precision.
Right.
So the older it is, the easier it is to measure because we have a lot of signal. Because we don't lose potassium essentially at a rate that's concerning. The potassium 40 is always there.
Okay.
But really young stuff, it's hard to measure because we don't have a lot of decay, so we don't have a lot of the daughter.
Right. So I'm not quite understanding why you have a better signal when it's older.
Because in this case, potassium is so abundant that it's the density of the daughter 40 argon. That's the harder part to measure.
Oh, okay.
Yeah,
okay,
yeah,
that
yeah.
makes sense.
Yeah.
Okay, yeah.
And yeah, the reason we're able to do it is because potassium and argon fractionate so much during eruption. Yeah.
Okay.
So then we have to ingrill the daughter again.
Right. So you're just like overwhelmed with the parent initially.
Yeah.
Okay.
Yeah, that's a lot of cases you have just excess parent, but the daughter's hard to measure. Some cases when you have younger, like if you're looking at uranium disequilibrium, so uranium and thorium has multiple steps before you reach a stable daughter.
Yeah.
Then those different intermediate steps can be limited by how much parent you still have left and your ability to measure that.
Right. Okay.
Yeah.
So basically this is one big like differential equations kind of problem.
Oh yeah.
Yeah.
That's everything always breaks down to.
Uh-huh.
And then of course you have to bring in nature, and so rocks just have so much variability in everything that it always complicates things.
Yeah.
Good conversation, you guys. Thanks for that, Kevin.
Thanks for asking.
Here's a question in the chat. Why are some fish in the deep sea, like the bathysaur, white? Wouldn't that make them easier to see in the dark?
It can. I think in their case it may be that they're often sitting out on sediment, and so they may blend in that way, or they may just not be that worried about it. So they may not be putting energy into creating that color for their skin. Because generating those chromophores is energetically expensive.
I imagine bathysaurs are kind of on top of the food chain down there to a certain extent. Hey, Kylie. If you were going back to get your master's in another field, or a field of science, what would you pick?
Me?
Yeah, based on all your experience now on the Nautilus.
Well,
He's fishing for compliments. Just don't say geology.
I'm just saying paramagnetism.
I was going to say, I should say paramagnetism. Right.
I'm still on an academic pursuit because I don't have my bachelor's. I just have my associate's degree.
Yeah.
So the jury's still out, really. I'm in a program for electrical engineering right now, but I could always pivot. I'm not so far into it where I couldn't just abandon ship and jump to geology.
Yeah. I mean,
I totally would though, yeah.
It would not be the lucrative choice. But,
Raj. The other thing about electrical engineering, I'm like, as a application that I'm interested in, or like a subsection of electrical, is like power and being able to, because I've like, in addition to like ocean stuff that I like, I'd really like to be able to work on providing like utilities for like as far as like emergency management situations go.
Yeah.
So I've been looking into like programs that incorporate some like power energy tracks within electrical engineering.
That sounds awesome.
I can't tell though if like the tracks really make a difference or just get the degree and find the job you want.
Yeah,
Both?
both. The internships you can do and research experience always helps along the way.
Right.
I find that networking is as key as anything.
Yeah.
But I think if you got a degree in electrical engineering, you're set for a lot of different fields. It's very...
That's the goal. Yeah,
Yeah.
that's the motivation for picking that discipline, even though I think I feel like it's a little bit really difficult.
Yeah. Thank you. That's a good answer.
Okay. Speed up? Yeah, that's the best it's gonna get. Yeah. No worries. I'll wait for maybe a second before I...
We're still continuing our ascent through the water column. We're at about 1,000 meters, 1,050. Hopefully we'll see some fish as we get into the shallower areas. Otherwise, snow. I wonder how far those lasers penetrate.
So I don't know about these ones, but before we were doing the Norbit thing, we had a sheet laser that we'd shine on the seafloor, and we'd do like a structure for motion, or not a structure for motion. What's it called? Whatever. You'd shine a sheet laser on the seafloor, and you take a picture from another angle.
Mm-hmm.
Structured motion, not structure from motion. That's another thing. Structured motion or structured light laser system. I got it. I know this thing. I went to school for this. Yeah, so the structured light laser system. So that we actually get ranges, and we could get clear pictures with the camera up to like almost 20 meters.
Oh, nice.
And at that point, like the laser is just like moves out of frame because it's not directly parallel with the camera frame. Otherwise, it wouldn't.
Right.
It wouldn't work. Yeah, but I mean, in clear water, it was crazy how far it could go. And that one spreads out over time, too. These lasers are focused. So the sheet laser actually, it's not quite 1 over r squared intensity loss because it's only spreads out in one dimension, but...
yeah, so at least 20 meters on these, then.
Yeah.
Cool.
The only difference is here is the power thing.
Yeah.
We were working with another lab. They had one of those sheet lasers on, and we were using up in shallow water in Rhode Island, which is sort of clear, but not like this.
Yeah.
But that laser was very powerful. They forgot to took the lens cap off the laser, and the laser, it didn't really mind. It just cut a hole in the lens cap.
Lasers will find a way.
Yeah. We're like, yeah. I was like, hey, there's like a little perfect slit in your lens cap. Did you just like do that to protect it? They're like, oh, no, we forgot to take it off. Yeah, that thing was awesome. That was like definitely wear your goggles with that thing around.
Yeah.
Just hang the lens cap next to the goggles as a reminder.
Yeah.
Totally.
Yep.
Well, the good thing about the sheet lasers is they do spread out. So they lose, burn through stuff intensity very soon. But
yeah. And I have a Jess-specific question. What's your favorite dancing type?
Oh, that's hard. There really isn't a favorite. No, I really like salsa dancing or tango.
Nice.
Those two are top favorites, I would say. Or give me a cumbia or something, I don't know, just anything that has a good beat.
Good.
Sometimes we see the crinoids that are in the water column and they look like they're dancing. They're having a good time.
They are graceful.
Yeah, little discotheque.
Mm-hmm.
Yeah, I don't think I knew about crinoids before this expedition, so I just looked them up, and so as juveniles, they stay attached to the rock, but when they're older, they'll swim. That's so cool.
That is very cool.
I sometimes get to see them while scuba diving, and it's always a treat. Under 1,000 meters to go.
Oxygen keeps dipping as we go deeper. As we go shallower, sorry. And it'll recover pretty soon. After about around 400 meters, we'll start to recover. Still no fish. Or jellyfish. Or squid. Or anything.
Do we know when our next dive will start?
Hopefully in around 4 or 5 hours, we'll see how Hook's hand is doing.
What other questions do people have for us?
Let's see. Oh, there's a question for you. What's your favorite place to visit in Boston? Public space.
Public spaces in Boston. Wow. There's so many good ones. Walking along Newbury Street is great.
Tales from the deep.
I like the North End in Charlestown. Boston Common, of course, and Beacon Hill. Boston's one of the great walkable cities, I think. You can kind of walk from one end to the other, and it's all accessible and clean and safe.
I've only been to Boston once, but I had a free day, and I think I walked almost 15 miles all over the city, just going different places.
Home of the Red Sox. Get 'em.
Oh yeah, Fenway Park, of course. Like, how could I leave that out?
Yeah, it used to be my dream actually to live in Boston. Like when I was younger, I used to create vision boards and like picture me living in like a loft or something. Yeah.
Have you visited?
I haven't actually. I haven't been to Boston. It's on my
To-do list?
yeah, definitely.
Yeah, great public transit system. Easy to get around. I lived up on the North Shore, in a town called Lynn.
Lynn, the city. I know the rest. Yeah.
You know the rest, huh?
Yeah, I'm from Fall River, and I went to school my first year out of high school at Salem State.
Oh, okay.
So, you know, pop around, get in trouble.
Gotcha.
Yeah.
Right. Up there near Saugus and Marblehead and
Swampscott.
North Shore.
Yeah.
Yeah, it's great. There's, you know, there are pretty amazing beaches up there, actually, which it sounds like it shouldn't really be the case, but yeah.
A lot of history.
Yeah.
Yeah. I thought that's what I thought I was going to do, like, out of high school, was, like, do cultural anthropology. Like, how did cultures decide what's important to them over time? And, like, the backwards and the forwards of that, like, the evolution of that.
Wow.
But I don't know. I just did a year there, and then I don't know what happened. But I didn't think I'd make any money doing that. And then when I went back to school, like in my mid-20s, I was like, it has to count. It was so hard to go back to school, right? Like once you're working full-time and like
Totally.
your life's already structured. I was like, it just has to like pay off, right? So engineering, and I had grown a little bit too to like realize what things in like everyday life I liked, like problem solving and like hands-on stuff, right? Not necessarily writing, like papers and things like that. So I was like, you know, maybe engineering, maybe that makes sense. Maybe engineering is that. It turns out.
Yeah, it turns out it
was.
is. Yeah, right.
Yeah.
No, that's a great story. Yeah, I think when a lot of people go to college, it's often because they sort of just didn't know what to do next. And it's sort of like, well, that's the next thing you're going to do. So, and I